Hello, everyone. My name is Glen Wickersham Garcia, and I am an undergraduate junior student at the University of the Sacred Heart in Puerto Rico. Being part of the NeuroID program has allowed me the opportunity to conduct research in Dr. José García-Arrarás Neurodevelopmental Laboratory at the University of Puerto Rico, Rio Piedras Campus, where we explore the regeneration of organs that our animal model, the sea cucumber Holothuria glaberrima, possesses. Although understanding this organism's abilities more in depth is of interest, it is not our only major purpose. Spread throughout the world, we have seen high numbers of patients with neurodegenerative conditions or cognitive dysfunctional disorders, such as Alzheimer's disease, Parkinson's disease, and multiple lateral sclerosis. An approach to understand how we can complement existing treatments is to contrast the molecular events that occur inside organisms that do possess this ability and don't form glial scars with those vertebrates that are incapable of regenerating the same complexes, such as us, right? To answer this inquiry, our laboratory has studied profoundly the morphological and molecular events that allow different tissue and cell compartments the ability to regenerate organs, such as the intestine after the sea cucumber eviscerates it as a defense mechanism. Like other echinoderms, we have focused our efforts in elucidating drugs that can intervene with these processes, or elucidating the messenger factors that are possibly being sent between the target cells and the receptor cells. Inside this broad question, I am in the works of developing two research projects. The first consists of the identification of neurotrophic factors and axon guidance restrictive, attractant, and permissive factors in the regenerating nervous complex of the mesentery, the organ that surrounds the intestine's luminal epithelium and the connective tissue. Some of the neurotrophic factors we've identified in the RNA-sequencing transcriptomes from both the intestine and the radial nerve cord are the NT3, with its respective receptor TRKC, and the receptor GFR alpha 4. These neurotrophic factors have been associated with molecular events such as modulating axonal acro, axonal branching and extension, cell survival, as well as microtubule stabilization. In humans, mice, axolotls, zebrafish, and in other echinoderms. Because during 7 days post-evisceration and 14 days post-evisceration, both deinnervation and reinnervation respectively are observed in the growing blastema and mesentery, it is hypothesized that the neurotrophins are secreted by the target cells in the blastema to the neurons so that these can then act accordingly to the stimulus, being there upregulated or downregulated in signals. The second project focuses on analyzing and predicting using machine learning models the behavior of regenerating neurons in response to these factor gradients, and how the presence of silencing these factors can influence the whole regeneration of neurons inside this complex muscular and epithelium environment.